Good morning, students. Now we can study here another type of hybridization. It is called as artificial hybridization. It is too much important for the crop improvement. Only the desired pollen grains are hand pollinated and fertilization is made. So we know that there are different types of plants. Here we are too much related with the crop improvement. So the plants which are used as a crop plant, in this type the artificial hybridization is done generally, and the fertilization is made by the hand pollination. हाताने त्याचं फर्टिलायझेशन केलं जातं. इमॅस्क्युलेशन म्हणजे रिमूव्हल ऑफ अँथर. वी कॅन से की अँथर हे मॅच्युअर होण्याच्या अगोदर फ्लॉवरपासून रिमूव्ह केल्या जातात, त्याला आपण इमॅस्क्युलेशन असं म्हणतो. इमॅस्क्युलेशन मीन्स द रिमूव्हल ऑफ अँथर before maturity takes place, and bagging also takes place. We know all these methods: emasculation, removal of the anther before the formation of pollen grain, it is emasculation. And after emasculation, if a polythene bag is kept on the flower, emasculated flower, to avoid the another entry of another pollen grain. So here we know that the artificial hybridization includes, it is handmade, it is important for the crop improvement, we can say, and this is followed by emasculation, and the bagging also takes place. Now here we have studied the artificial hybridization. Now we will go to the double fertilization. We know that double means 2 times, so it is an important character of the angiospermic plant. Angiospermic means we know that whenever ovules are enclosed in the ovary, or we can say the seeds are present inside the ovary or fruit, so it is called as the angiosperm. Now what is double fertilization? We can study here that it is discovered by Navas-Chin in the plant Lilium and Fritillaria. The scientists who had made research in this double fertilization is Navas-Chin, and the plants they are used, they are the Lilium and Fritillaria. Now when the pollen grain falls on the stigma, then it starts germination. After falling the pollen grain on the stigma, it starts germination. We have studied that when the right type of pollen grain is there, it will germinate there, and a wrong type of pollen grain cannot germinate. So when the pollen grain falls on the stigma, then it starts the germination. The pollen tube is formed. We know that inside the pollen grain there are germ pores, small pores are there. So from the pollen grain, germ tube is produced, which enters in the style. We know that there are different parts, stigma, style, and ovary, that is present in the carpel. So pollen grain germinates, it enters in the style, and from the style to ovary chamber, it reaches to the ovary chamber. Now the growth of pollen tube is guided by the chemicals secreted by the synergids. We know that synergids are present inside the embryo sac. 2 synergids and egg is present toward the micropylar end. So whatever it may be the chemical secreted by the these synergids, the growth of pollen tube takes place. Now the pollen tube enters through the micropyle. The process is called as porogamy. See here there are different terms. Porogamy means entry of the pollen tube through the micropyle. Now in some cases it enters through the chalaza, that is called as the chalazogamy. So there are some variations. Whenever the pollen tube enters through the chalaza, it is called as chalazogamy. When it enters through the micropyle, it is called as the porogamy. See here we are inside the double fertilization. It is discovered by the Navas-Chin. It starts when the pollen grain falls on the stigma, then it produces a tube. The tube enters through the style and into the embryo sac. The entry of pollen tube through micropyle is called as porogamy. In some cases entry takes place through the chalaza, it is called as the chalazogamy. Now see here, through the micropylar end, it reaches to the embryo sac. Where it reaches? It reaches to the embryo sac. And the watery content of synergids are absorbed by the pollen. There is a watery content present. It is absorbed by the pollen tube. It ruptures and two male gametes are released. Here the gametes are two, and the two male gametes, they are released. See here, the entry of pollen tube through the micropyle, it is called as porogamy. The pollen tube absorbs some chemicals or watery content from the synergids, and its breakdown, it can rupture, and the two male gametes are released. How many gametes are present here in male gametophyte? Two male gametes. They are top-shaped male gametes. They are released. These gametes are non-motile and are carried in pollen tube, so it is called as siphonogamy. See here, there are different terms. When the pollen tube enters through the micropyle, it is called as porogamy. When pollen tube enters through the chalaza, it is chalazogamy. When the non-motile gametes are carried in a pollen tube, it is called as siphonogamy. It is called as siphonogamy. Now the union of one male gamete, now here gametes are two. Union of one male gamete with egg is called as syngami. There are different names. Syngami, union of one male gamete, that is n in number, haploid in number, unites with the egg, it is also haploid in number, and the process is called as syngami. And due to the union of these both, that is one egg and one male gamete, diploid zygote is produced. Zygote is diploid. See here, union of another male gamete. Now one male gamete is remained. The union of another male gamete with the, we can say, secondary nucleus. With the secondary nucleus, it is called as triploid fusion. Why triploid fusion? Because male gamete is n in number and the secondary nucleus is 2n in number. So union takes place, this is 2n, and the triploid endosperm is present. So fusion takes place 3 times. So that is double fertilization. Fertilization takes place 2 times, one with the egg, another with the secondary nucleus. So double fertilization. And triple fusion means one male gamete and 2n, that is the secondary nuclei. It is called as the triple fusion. The triploid endosperm is formed due to this union, because in the life cycle, this triploid endosperm, it is 3 in number. Gametes are haploid, original plant is diploid, but the endosperm is triploid, which is nutritive in the nature. Now zygote develops into embryo. Zygote develops into embryo, and endosperm is formed by the union of gamete with the secondary nucleus. Now see here how it is formed. We have studied. Endosperm is 3 in nature. It is nutritive. तो 3n नंबर्स आहे. ओरिजिनल प्लांट डिप्लॉइड आहे, त्याचे गॅमेट हे हॅप्लॉइड आहेत, पण त्यापासून तयार झालेला एंडोस्पर्म मात्र हा ट्रिप्लॉइड इन नेचरचा आहे. सिनगॅमी इज अ युनियन ऑफ generative fertilization. Syngami is a type. Which type? It is a type of generative fertilization. And the triple fusion, it is a type of vegetative fertilization. We are getting here the differences. Syngami means union of one male gamete with the egg. It is the generative fertilization. And fusion of one male gamete with the secondary nucleus, 2n in number, it is a type of vegetative fertilization. Now, double fertilization and triple fusion is a character of angiosperm. जितक्या झाडांच्या बिया ह्या फळाच्या आत असतात त्याच्यामध्ये डबल फर्टिलायझेशन आणि ट्रिपल फ्युजन हे एक महत्त्वाचं कॅरेक्टर मानलेलं आहे. सो डबल फर्टिलायझेशन and triple fusion is the character of angiosperm. Now see here, it is the stigma, this is the pollen grain, it produces the pollen tube, it enters through the style. The process we know, it is called as the porogamy. Here it enters in the through the micropyle in the embryo sac. Now here two gametes are present, At lower side, egg and two synergids are present. At middle side, here we will get the secondary nuclei, and at the upper side, here antipodal cells are present. They are 3 in number, 3n in number. Now one male gamete among the two unite with the egg and form the diploid zygote. Remaining unites with the secondary nuclei and form the triploid endosperm. It is the double fertilization and triple fusion in the angiosperm. Now see here, what are the, we can say, significance of double fertilization. So significance means nothing but what happens, what is, for what it is useful. It is the character of angiosperm. See here, it is the character of angiosperm. We have to write here the 4 points. It is the character of angiosperm. Diploid zygote develops into embryo. Diploid zygote develops into embryo, which develops into new plant. So zygote is responsible for the production of new plant after formation of seed and germination. Now the triploid endosperm, that is the primary endosperm nucleus, develops triploid endosperm. It produces the endosperm. It is a triploid. It is nutritive in function. See here, diploid zygote develops into embryo, which develops into new plant. The triploid endosperm, that is from the primary endosperm nucleus, develops into endosperm, which is nutritive in function. Now it restores the diploid condition by fusion of one male gamete, that is n in number, with the egg n number and form the diploid zygote. The original number of plant is diploid. So from the haploid male, haploid female, diploid number is maintained, and it helps to avoid the polyembryony. Polyembryony cannot take place. Here these are the significance of double fertilization. What are these? Diploid zygote develops into embryo, which develops into new plant after development of the seed and after getting favorable condition. The triploid primary endosperm nucleus develops into triploid endosperm. It is nutritive in function. It restores the diploid number, that is the fusion of male gamete n number with the fusion of n, that is the egg, and form the diploid zygote, and it helps to avoid the polyembryony. Thank you.